Okay. Konnichiwa. Thank you for coming today. So today, the idea of this session is to give you all a better idea of how to do stable isotope labeling experiments, the power of it, and also to try and make you less scared of approaching it. This time last year in Niagara Falls, the Metabolomics conference there weren't that many stable isotope labeling talks given over the course of the week, and so the three of us decided that maybe we need to educate some people on how it can be useful for their projects. It's really something that you don't need to be scared of. So in this talk, what we're going to do is we're going to go through what stable isotope labeling is, how it can be powerful, and we're going to give you some case studies or examples of how you can use it in your work. We're going to try and keep it quite simple, but as the talk goes on, it will get a little bit more complicated. But please don't be scared. We think it's still going to be quite easy. So because this is day one of the conference, just a quick reminder of what metabolomics is. So when we're doing metabolomics, we're always trying to look at as broad a range of low molecular weight molecules as we can in any given cell, tissue, organ, biosystem, biofluid. And I'm giving you this example here, this little cartoon. This will be important later. This is an example of what I've done in my past research in the malaria parasite. So when I'm doing work with the malaria parasite, we're looking at Plasmodium falciparum. We're looking at Plasmodium falciparum cells that reside within the red blood cell. I'll talk more about that later, but I just want to use that as a little analogy here. And when I do metabolomics with malaria, I'm looking at the metabolites that are in Plasmodium falciparum. I'm looking at the metabolites that are in the surrounding red blood cell. I'm looking at those metabolites that are taken up into the parasite. And I'm looking at unidentified or unexpected metabolites. Now, so far, that all accounts for maybe untargeted metabolomics or discovery metabolomics. But this talk is about another technique, which is stable isotope labeling. And in this sort of experiment, what we do is we choose a carbon source or a nitrogen source or another source, and we use a stably labeled version of that, in this case, carbon-13 glucose. We add it to our cells or our organs or our organisms, and we see where that stable isotope goes. This is a very powerful technique because it can give us an idea of the precise activity of what's going on in the cell at that specific time. So this is just a very brief overview slide of the difference between targeted and untargeted studies. The vast majority of the talks this week are going to be profiling, discovery, untargeted, call it what you like, that kind of metabolomics. And that's great. There we're trying to look at as many metabolites as possible. We're trying to get a broad idea of what's going on in our samples. But sometimes we can miss some of the nuances. Simply knowing abundances isn't always enough. What we like to do is go into a targeted approach, but not only just that, not just highlighting a specific pathway or pathways, but we want to know what is the activity of those pathways. And the way we do that is by using, as I said, stable isotope labeling. This is really important because we can zone in, we can get very intricate information, we can find out new things about metabolic pathways that we might not have known before. But we always have to be mindful that when we're doing such a study, we're really being shining the flashlight right at what we want to look at, so we might miss a whole range of other things. Targeted metabolomics isn't the only answer, but it can be used with your untargeted metabolomics approaches to get a holistic idea of what's going on in your sample type. We're looking at a whole range of metabolites, of course. They can vary in size, polarity, volatility, etc. LCMS is great. You can see thousands of features in any one experiment. GCMS is perhaps less used. We just saw Oliver Fien walking out of the room before. I hope that wasn't an indictment of what he thinks about this presentation. But he's one of the grandfathers of GCMS metabolomics. And many people still like to use this, and the first part of the talk will be focusing on GCMS, mainly because it can be very useful when you're interested particularly in energy metabolism. It's great for sugars, it's great for the TCA cycle, it's great for amino acids, and so I'll focus on that. We're not going to talk today about NMR, but this is another very common tool in metabolomics, and it's also very useful for stable isotope labeling studies as well. But we don't have time to discuss that today. So why do I use GCMS? As I've just said, it's very powerful for looking at some specific pathways. Central carbon metabolism is definitely one of those. This slide here just shows some of the metabolites that you can see, or the metabolite classes that you can see using GCMS that you might not be able to see using LCMS, and vice versa. And there's a lot of overlap in the middle. And, you know, I've put sugars here. Sugars can be seen by both GCMS and LCMS, but in general, it's much better to identify your sugars using GCMS because you can get much better separation power. So we use it quite a lot. It's also, by the way, if you don't use GCMS, it's cheaper, it's more robust, you get prettier peaks. And again, as I said, you get some really good separation where you might need it. It's quite a good starting instrument for anybody trying to do metabolomics for the first time. And here is a GCMS. And this is my cartoon. I made this slide in 2001, and I'm trying to keep it going as long as possible, so I'm going to use it again. So when we're doing GCMS, we add our sample, which is in a liquid form, and we immediately volatilize that to 270 degrees. And then it is carried on a carrier gas, that's why it's called gas chromatography, onto a very long, 30 meters is typical, column that is coated in a hydrophobic gum. That's sitting at usually around 70 degrees, and your metabolites that were volatile, in the gas phase, will condense onto that hydrophobic gum. Then what we do, and here's the mass spec at the end of the GC. Generally in GCMS there are two kinds of ionization. First one is chemical ionization, but far more popular is electron impact. And I'm just going to show you how we do that in a second. So what happens, as I said, your sample is injected onto the column, it condenses onto the column, and then we raise the temperature of the oven. So we're going typically from around 70 degrees or 80 degrees up to 320 degrees. And at any given point, your metabolite will come off of that hydrophobic gum and then be eluted off of the column into the electron impact ionization chamber. Here it's bombarded with electrons, generally producing positive ions, and those just go through a quadrupole into your mass spec of choice. We use mass selective detectors, quite basic really, but it's still good for what we want to do. As I mentioned earlier, we get some very good separations with GCMS. Generally, smaller molecules will come off first, more polar ones will come off first, more larger ones and less polar will come off later. But importantly, even though mannose and glucose, for example, are the sugars of the same size and empirical formula, because of the position of their OH groups and the subsequent derivatization processes that will be discussed more later on in the presentation, we can get some really nice separation based on the spatial orientation of these derivatization groups. Okay. So what does a GCMS chromatogram look like? Well, it looks like this, as I said. This is much nicer, sharper peaks than you'd get in a standard LCMS chromatogram. And while in an LCMS peak you might see tens, hundreds of metabolites, in a GCMS trace you'll usually see a few, maybe half a dozen at most. Sometimes it's just one metabolite. And I've highlighted here myoinositol. I know that's myoinositol because when I look at the spectrum at that point, I see this characteristic spectrum pattern here that I know is the fragmentation pattern of the derivatized molecule. Electron impact is always the same. It's the same today as it was 2 years ago, as it will be 2 years in the future. So we always know when we're looking at our metabolites that we're going to see not only these fragment ions, but in this specific ratio. Now, when we do stable isotope labeling experiments, what we're looking for is the incorporation of, for example, carbon-13. So in this case, what I'd be looking for is a conversion of these 318 ion and this 305 ion. I would expect that to go up according to how many carbon-13s are incorporated. With the 318, we've got 4 carbons here. With the 305 ion, we've got 3 carbons, and so on and so forth. So when it's labeled, what we'd expect would be to see the mass of 305 going up by 3, because that carbon-12 has turned to carbon-13. 3 carbons, they've all gone up by 1, so it's going to be 308. Similarly, this 318 should go up to 322. And this cartoon here shows you the sort of thing we'd like to see. In this particular instance, what we would glean from this is that there's maybe, I don't know, maybe around 40% of all of the myo-inositol here is labeled. And that's pretty simple. But it does get more complicated than that. Excuse me. So in this cartoon, I'm showing you citrate. And when we're looking at citrate, citrate's got 6 carbons. So if it was fully labeled, we'd expect to see a +6. So in this particular cartoon, what we see is that we see, oh, my pointer's gone. I think the pointer's like, oh, there we go. We see that around a quarter of these molecules are unlabeled, no label incorporation, and around 3/4 of them are fully labeled. Okay, and this is essentially a cartoon of what we're seeing. So 75% of these molecules are labeled. But what about this situation? Here, again, about a quarter of them are unlabeled, and in this case, 3/4 of the molecules are labeled, but only in 2 positions. But we would still say that 75% of these molecules are labeled. And similarly here, we might have a mixture, where some have got 2 carbon-13s, some have got 4 carbon-13s, and some are fully labeled. Of course, all of these cases, 75% of the molecules are labeled, but the number of carbons labeled, or the fraction of carbons labeled, is different. In this first case, 75%, here only 25%. Oops, oh, sorry, got a bit ahead of myself. Here only 25%, and here around half of the carbons are labeled. This is why it's really important when you look at publications and you see a heat map that generally will show you the percentage of molecules labeled. There might be quite a lot of information that is hidden from view, and maybe you could go in deeper and find out more about the system. And we're going to talk about that now in the next section. So here, I've already talked about the malaria parasite, and I'm going to give you a bit of a case study here on how I've used these kind of techniques to learn new things about the malaria parasite. So just a very brief overview. I want to try and give this talk a biological context. Malaria, I'm sure we are all aware of malaria, and it still, even today, affects many people around the world, especially the young. There are drugs available, but there's always drugs resistance that gains traction year upon year. That's why we need to use different drugs in different regions of the world. And 2024 is a big year for malaria. There's a vaccine that's been rolled out, but this doesn't really negate the requirement for drug treatments for those who are already infected and for those for whom the vaccines may not work. So we still need to study this organism. So this is a complicated diagram, but it's important in the context of telling you the cell types that I'll be talking about. So just to go over this very briefly, when we get bitten by the mosquito, the parasite is in this form called the sporozoites. That immediately invades your liver cells. It stays there for a short amount of time, and then at some point they rupture out of the hepatocytes and enter the bloodstream. This is the important stage of the parasite, really, because that's how we get the disease. When you suffer from malaria, it's because it's in your red blood cells. Okay? And we've got this complicated lifestyle here, asexual life cycle here called schizogeny. And today I'll be talking about these later developed stages here, these schizont stages. This is where your cells are juicy and full with a lot of parasites. Once they reach a certain number within any given red blood cell, they rupture. Most of those parasites will then reinvade in continuing the asexual cycle. but a very small percentage of them will change into the sexual stages. And it's these sexual stages that I'll talk about a little bit later that are responsible for the proliferation of malaria. They're the ones that are taken up by the next mosquito bite, where they reproduce in the mosquito midgut and continue the cycle. But for now, let's just think about these asexual stages, okay? So what's interesting about the malaria parasite is that for years and years and years, it was expected that they got all of their energy, all of their ATP, only from glycolysis. The reason for that is because if you're going to use the more efficient TCA cycle for generating energy, you need and absolutely need pyruvate dehydrogenase. Now that sits in the mitochondrion along with all the TCA enzymes, but in the malaria parasite, pyruvate dehydrogenase is there, but it's in this other organelle called the apicoblast. I don't have enough time to talk about the apicoblast today, but essentially it's well away from where it's needed to be for the TCA cycle to be active. But we wanted to investigate this a bit more, and so we started to do some labeling studies. Now here, what we've done is we've labeled our parasites, our asexual parasites, for about 48 hours with carbon-13 glucose or carbon-13 glutamine. Glucose gets into the TCA cycle going down glycolysis and then being converted to pyruvate, and I'll talk about this a little bit more later. Glutamine is a bit of a more direct route. It goes straight from glutamine to glutamate and through to alpha-ketoglutarate, okay? So when we're looking here at the label incorporation, we can see that there's a lot of label incorporation, as we can see from these hotter colors. There are more molecules labeled here in these lower metabolites of glycolysis. But when we look at the TCA metabolites, we can see that it's nowhere near as labeled. But it is labeled, and this was quite important in and of itself. Before, we didn't think that there was any label incorporation, but even though it's not a great deal, again, this is 48 hours, we are seeing label incorporation. And we can see as well that when we're labeling with glutamine, we're seeing a lot more glutamine incorporation, much more of an easy route to get in this way. So this was interesting. But I've already told you, you shouldn't believe heat maps just by looking at them. So we wanted to look a bit more. We wanted to look at the isotopolog patterns. We wanted to see how many carbons were incorporated and how those carbons were distributed in each molecule. So here, what we've done, again, it's the same experiment. This isn't a new experiment. We've added that carbon-13 glucose, which has got 6 carbons. By the time it gets down to PEP and pyruvate, there's 3 labeled carbons. When that crosses into the mitochondrion and is converted to acetyl-CoA, that's 2 labeled carbons. So if you're going to go around the TCA cycle once, each molecule will have 2 labeled carbons, okay? And of course, if you go round again, you're going to take up another 2 labeled carbons, so you'll start to see 4 labeled carbons. And then if it goes around again, you're going to see the fully labeled, the 6-carbon citrate. Interestingly, there's another route in as well. This depends on your organism, but this is the way it happens in Plasmodium falciparum. We can get carboxylation of PEP as well. So here, this PEP will pick up an unlabeled bicarbonate from the cytosol to form oxaloacetate. In this case, it's got 3 labeled carbons. That can be converted to malate, and malate can cross into the mitochondrion and contribute to the TCA cycle and energy generation that way. Okay? So what does this look like? Well, these are the real results. And what you can see, if we maybe start looking at citrate, we can see that over these 48 hours, we're seeing that +2 I was telling you about, we're seeing +4, and we're seeing some +6, okay? We're also seeing +3 and +5, quite a lot of +5 as well, which suggests that both of these routes of entry are quite active. So we're seeing a lot of pyruvate to acetyl-CoA, and we're seeing quite a lot of this PEP to oxaloacetate to malate and the crossover into the mitochondrion. When we do the reciprocal experiment with glutamine, again, we're going to see a similar thing. So the glutamine has got 5 carbons. When that gets into the mitochondrion as alpha-ketoglutarate, that's also got 5 carbons. Then it loses a carbon to form succinate, so we're going to have +4. We're going to have +4, +4, +4. But, counter to what I just told you about glucose, when we're doing this kind of experiment and we have unlabeled glucose, we're going to see an incorporation of +2 unlabeled carbons in the form of acetyl-CoA. So actually, the more the TCA cycle goes around, we might see less labeling. So what does that look like? And we can see it here. So most of the molecules are +4, but over this 48-hour period, we're seeing some +2 as well, and that's because of this incorporation of the cold acetyl-CoA carbons. Okay? So in one experiment, we were able to determine that the labeling pattern was indicative of there being a canonical TCA cycle. That's not really big news for most organisms, but this was absolutely thought not to be the case in Plasmodium falciparum. So that was quite interesting. Is it interesting? Well, we wanted to see whether it was interesting by seeing if it had any kind of effect on the malaria parasite itself. So what we did here was we inhibited the TCA cycle using sodium fluoroacetate, which blocks the conversion of cisaconate to isocitrate. I haven't shown those 2 metabolites in this simple picture. But what we do see, when we inhibit the TCA cycle in this way, you can see a huge amount, this isn't labeling now, this is abundance, you can see a huge abundance of citrate. It's building up. Now that wouldn't happen if the TCA cycle was going in reverse. It wouldn't happen if there was no TCA cycle. It would only happen if it was working in a canonical direction. Now, in blue here is the asexual stage of the parasite that I was telling you about, but just by chance, we happened to look at the sexual stages for a completely different study, and we saw that there was even more citrate built up in the sexual gametocyte stages, and we thought that was interesting. So we investigated that later. Before we did that, though, we wanted to know, does blocking the TCA cycle in these asexual stages have any effect at all? To do that, we just cultured them and we looked to see if there was an effect. And here, we just grew our cells normally. Generally, when we're doing red blood cell culture of malaria parasites, we're looking at around 12% labeling, 12% infection. We don't want to go above that because the parasite will crash out. So 12% in this case is the maximum. And as you can see, unfortunately, when we added our inhibitor, there wasn't really any kind of significant difference, which was a bit disappointing. And this is just a control here. But we thought there might have been something interesting in the sexual stages, so we wanted to investigate that further. And here's the same result I showed you earlier. It's exactly the same. And in this case, this is the labeling pattern that we saw when we repeated the experiment, but in those sexual stages. Now, what you can see here is 2 things, 2 important things. First of all, look at the heat of these colors here. We're seeing much more label incorporation. A lot of incorporation into these TCA cycle intermediates, and much less from glutamine. So there's been a carbon source preference switch. So we know immediately that during these sexual stages, for whatever reason, that stage of the parasite prefers to use glucose. But the other thing is down at the bottom here. I mentioned earlier that we have to do a 48-hour label to get this kind of incorporation into these asexual stages. But this was only a 4-hour label. So not only is there a carbon source switch, but the rate of incorporation has hugely gone up, suggesting again that the TCA cycle is much more active in these stages. Again, I'm going to look at these isotope log patterns again. I want to see how the carbons are incorporating carbon-13. And just looking here, maybe focusing on these left ones. Now, this might look complicated, but it's actually the same as the bar charts I showed you earlier, but as a time course. So I just said that, again, let's maybe look at succinate is probably an easy one to look at. So we can see here that we're seeing maximal labeling. So this is all 4 carbons of the succinate are labeled by 4 hours. We knew that from the previous heat map I showed you. But this tells us that actually it's much quicker than that. Maybe up around an hour or even 2 hours, we're seeing that maximal labeling. But we can see some other interesting things here. We can see that the +2 goes up very quickly, even after 20 minutes. We're seeing that first round of TCA cycle activity coming in, and then obviously that will drop off as the +4 takes over. And again, just like the sexual stages, the pink line here is the +3, so we know that we're getting stuff coming through this route as well. Okay? So what this tells us in a trice is that the gametocytes, these sexual stages, complete carbon source switch and a huge increase in activity. But does it matter for these stages? So I'm just going to finish up with a little bit of biology now, and these are what the sexual stages look like. So they're morphologically very different. They're very easy to detect under the microscope. And when we were looking at these, what we wanted to do is to see what is this progression like. And this usually takes about 10 days or so. And so what we did was we looked at the abundance or the percentage of cells that were infected with these early ones, and then those that had been allowed to mature to the later stages. Very easy to tell. But this is a control graph here. What you can see is that after 7 days, yes, there's been a parasite drop-off. That's normal in our cell culture regimen. But by 7 days, we can see that almost all of the parasites are these nice, mature parasites that are ready to be taken up by a mosquito and complete the life cycle, complete the transmission. When we add sodium fluoroacetate, that TCA cycle specific inhibitor, what we can see is that not only is that parasitemia drop-off very great, but hardly any of them that get to maturity, hardly any of them get to maturity by day 7. And actually, I'm not showing it here, but when we look at these under the microscope, these really look quite ugly and they don't look healthy at all. Okay, so that's interesting, good. We think that the TCA might be important to these cells, especially in the sexual stages. But we were left with a bit of a mystery, which is, I had told you at the start, there is no pyruvate dehydrogenase. And I could give another 40-minute talk about our investigation on what could be going on here. But to cut a long story short, we suspected that branched-chain ketoacid dehydrogenase was a likely candidate. And so we knocked it out in a related parasite to Plasmodium falciparum, Plasmodium berghii. And here we could see that when we knock this out, we're seeing a lot less label incorporation into these TCA metabolites when we're using carbon-13 glucose. But there wasn't really much difference when we were using carbon-13 glutamine. So that's interesting. That suggests that, yes, BCKDH might be acting as it. And this is one of the things I mentioned earlier, that GCMS has got maybe a limited number of metabolites that it can detect, maybe in a few hundreds. One of the metabolites that it can't detect is acetyl-CoA itself. So this is the only LCMS slide that I'll present. Bart will talk a lot more about it shortly. But here we turn to LCMS, and here in the wild type, we've got our label incorporation, and you can see this +2 acetyl-CoA, so we know that it's labeled. But when we have the knockout, there's a complete ablation of the labeled form. So that told us that BCKDH indeed was acting as PDH in these cells. Okay, how am I doing for time? Not too bad. Okay, so just to finish off again with a tiny bit of biology, we wanted to see, does this BCKDH affect transmission like I just suggested it might? So just quickly, once they get into the mosquito midgut, they form microgametes and then macrogametes. Then they form these things called ookinetes, and then you get a mature oocyst. And if you don't get a mature oocyst, you don't get transmission. So what we see here, again, maybe just concentrate on the dark blue and the red. When we knock out this BCKDH, when we knock out the TCA cycle, what we see is a great reduction in the ability of micro and macrogametes to form, gametocytes to form. We see much lower oakinate conversion as well. And importantly, we can see here that we get almost no oocyst conversion. So that gives us great confidence that transmission itself is likely to be inhibited. And indeed, since we did that work, which was quite a few years ago now, there's been many studies that have been investigating TCA enzymes and related enzymes. And you can see here that in a lot of these knockouts, we're seeing almost a complete ablation of formation of oocysts, and we can see that it shows that they could well be good drug targets for stopping transmission. Okay, well, I'm going to leave it there and hand over to Bart, who's going to tell you about similar studies, but using LCMS. And that, so that was the easy section. So I hope that was all clear. Things get a bit more complicated now.
Good afternoon. Just a short recap on mass spectrometry. So there's always two parts. We have a separation part called the chromatography. So as James pointed out, this is you can have gas chromatography, but I will talk about the liquid chromatography. Why do we need this? We need to separate molecules before they go onto the actual measurement, the mass spectrometers, because mass spectrometers cannot deal with too complex of samples. If I would skip the chromatography, I would actually see only a couple of metabolites. And this mainly is due to the fact that they compete for ionization. And of course, when a metabolite turns into an ion, that's actually when we can detect it with mass spectrometry. This is a typical liquid chromatography mass spectrometry setup. So we grow cells, in this case adherent cells. You can do it for suspension cells as well. We grow them in a 6-well plate, and typically we have about 300,000 cells per well. Why is there one well in red? This is a practical thing that you should be aware of. It's because of this. A typical 6-well has this corner cut off. We do see more evaporation there. So if you want to compare like a medium from this well with another, you will see that you have actually differences, and this can easily be 10 to 20%. Just because after 24 hours there's a significant amount of the medium evaporated from this well. So when you run an experiment in a 6-well, don't use this one, especially not for metabolomics. For tracer metabolomics, we actually use labeled medium, so we're going to replace one of the nutrients in the medium by its 13C analog. This can be any sugar molecule, glucose, galactose, mannose, but also amino acids, fatty acids, we can provide to the medium as a labeled source. This being said, only add one source at a time, because if you add both glutamine and glucose 13C labeled to a setup, it's very difficult after your analysis then to pinpoint what caused the incorporation of the 13C atoms in a certain target. What's very important here is that we have 3 sets where we have 13C medium with cells. And then, of course, here, I think this is the most key setup ever. It's your unlabeled setup. Why is this one important? Because if we look for incorporation profiles, we have to be sure that there's no circumstances that trigger a certain mass to pop up and actually to create like a false profile. I had just one example in my career where people had a brilliant incorporation in serine. They saw a 13C incorporation that made sense from a biochemical point of view, but they never actually added this setup. And it turned out that the compound they added gave an analog that was very similar, I mean, less than 1 ppm similar to the incorporation in the serine. And it also eluted at the same time, so you can get unlucky in research. But that actually caused a PhD student to become very depressed after 2.5 years of their research. So keep in mind, it's just one additional setup, but it's so essential when you do tracer metabolomics. Of course, the labels that we add, we initially aim to go for steady state, meaning that we look at a balance between the incorporation of 13C atoms from our nutrients with all the other sources that can contribute to that specific nutrient. So typical incubation times, glucose 24 hours, amino acid 48 hours, and free fatty acids, we go up to 72 hours. Of course, we then extract, and I think this is the main advantage of LCMS-based tracer metabolomics. You remove the medium, you wash the cells, you remove the washing solution, you add the extraction buffer, and that's it. That extraction buffer you can immediately load onto your mass spec and do the analysis. So there's no need to dry the sample, derivatize the sample, and so on. Of course, this is real life. There's a disadvantage with liquid chromatography. The separation is not that beautiful, meaning that where we have very sharp peaks with a beautiful baseline, in liquid chromatography, it's the nightmare for the data analysis and integration thereof. Keep in mind, there's many flavors of mass spectrometry. There's no golden setup. If that would be the case, we would all be using the same one. There's many setups, unfortunately. Each setup is designed for specific targets of metabolites, and it's really up to you to upfront know, I want to study these metabolites. This is the ideal setup. Liquid chromatography, this is a schematic of it. So instead of the gas or the helium that James talked about that act as the mobile phase, we are using liquids. In the majority of cases, you should have 2 bottles on top of your chromatograph, A and B. And actually what we do is we load the analytes in a certain unique condition, let's say pure solvent A. After this loading, we gradually switch to solvent B. So what will happen over time, the molecules that are bound to the column, to the stationary phase, will gradually elute, and they are pumped to the detector or to the mass spectrometer. Typical liquid chromatography setups are reverse phase. Don't memorize this, but I think the most important thing here is this, the mobile phase. So here with reverse phase, we go from liquid, from a polar liquid phase to an apolar liquid phase. This has repercussions. Depending on the molecules or the metabolites you would like to measure, imagine you want to measure very apolar molecules. It's not ideal because you start with the polar phase, meaning that they will be stored in your mass spec file in a very polar phase. So they will probably precipitate. But vice versa, if you want to look at amino acids, these dissolve very well in water, so they easily go onto that. So everything has repercussions. Reverse phase is prone to bind more hydrophobic molecules. There's a bypass, you can do ion pairing. And now I have to warn you, when you do ion pairing and you're not familiar with it, you will put your instrument lifetime on ion pairing, meaning that the molecules that we add, they really improve the separation of, let's say, polar molecules onto the reverse phase, stationary phase. However, you contaminate the complete system. To give you an example, I told a colleague I had beautiful results with tributylamine. He tried it out and he thought he could go back. It took him 2 years. 2 years of cleaning the machine before he actually had again a bit of good results. So it means that once you go ion pairing, you stay with ion pairing. Also keep these machines away from the others because the ion pairing agent goes into the air and it also pops up sporadically on your other instruments. HILIC is the most popular LCMS type of chromatography because this one is a bit of everything. And that's also the one that we will use most when we do typical LCMS experiments. The liquid phase goes from apolar to polar, and then the stationary phase is polar, so it does bind amino acids, nucleotides, and so on. At the end of the day, this is a typical chromatogram, and then it shows the ion trace of ATP. Keep in mind, we're looking at ions, so this is negative mode, so we subtract the hydrogen, we add up an electron, and that is going to give us the exact mass of ATP. As you can see, the elution is much broader than in GC, so this is not 5 to 6 seconds anymore, this is about a minute, but that also gives us the opportunity to connect high-resolution instruments that need more scan time for tracer analysis, because the broader the peak elutes, the more chance the mass spectrometer has to scan, and the more chance it has to collect ions. At the end of the day, you get very beautiful, very accurate mass measurements. So this is ATP. If we then zoom in, and this then goes into the tracer field, instead of regular metabolomics, we're not going to study one molecule anymore, or one mass. Every molecule that we want to look at is going to be looked at from its carbon point of view. Meaning that for ATP, I have 10 carbons, there will be 11 masses to follow. Every isotope, every 13C isotope, we have to quantify, we have to measure. So yes, it's more work for tracer metabolomics, but it does pay off at the end of the day. So if we zoom in the ATP, this is the typical 1 plus 1.00335, and then again plus 1.0335, and we're actually going to add that into our library. Are we doing this manually? We used to. That actually caused depression with many people in my lab. Luckily for us, there's tools out there like LMaven, where we now can overlay all the samples. We look up the specific targets, and then we actually let the software extract all of the isotopes that we need to look at. So there's different stuff. There's a name and a mass range. There's a retention time. So this is library-based, looking up of metabolites. At the end of the day, this is what you get, unfortunately. And I think David will talk a bit more on visualization, but this is still what we do. What you see in the columns are all the isotopes per metabolite. And then, of course, here the rows are the actual samples. What you see are percentages, and that's actually what we do. We interpret these and we try to figure out what's going on. With LC, we can actually look at, and this is now the majority of my talk, we can look at a special type of molecules. Large molecules, small molecules. Think of nucleotides, ATP, sugar nucleotides, UDP glucose, UDP glucanac, and so on. These are, from a tracer point of view, very interesting. And I call them composite molecules. Why? Because they are actually built out of different units that come from specific pathways. So if this would be a composite molecule, and you can see the different parts, these are actually coming from pathways. So there's a nutrient coming in, it goes via pathway B, A, or C, and certain intermediates of this pathway B are actually being used to build that specific composite molecule. When I add a layer of tracers, you actually will see why this becomes interesting, because pathway A, think of glucose and then glycolysis, is heavily relying on this nutrient, but it's going to incorporate this subunit into its composite molecule. Same for B, same for C. What this means is that if I look at a composite molecule from a tracer perspective, I actually have the information of 3 pathways without the need to look at all the different intermediates of this pathway. This is one of my favorite molecules because it's linked to at least 4 pathways. It's UDP glucanac, but we can do the same thing for different kinds of these composite molecules, ATP as well. The colors actually show you the different subunits. So in yellow, this subunit is actually coming from the hexosamine biosynthetic pathway, actually glucose going off from glucose or fructose 6-phosphate going in there. Same for the acetyl group, that's lower glycolysis, the ribose group, and then the uracil group. Each subunit comes from a pathway. So this is the idea that we had in the past, was like, if we would only look at this molecule, is it less work for us to do the tracer metabolomics? And also very interesting, if we actually now dynamically look over time, look at the different subunits and how the tracer is incorporated, can we actually tell something about kinetics? So the reason we actually came up with this was the fact that at our institute back then there was a big shot PI that said, like, I have the molecule that blocks glycolysis. We had certain doubts because when we used the molecule in not all systems it was actually blocking glycolysis. So we decided to set up an experiment. So we grew cells. These were endothelial cells. UVEX. We grew them in glucose, and then we checked different compounds. So these are parallel setups. So we had compound X, so the unknown compound that should block glycolysis. And then we had 2 doses of 2-deoxyglucose. This is an established glycolytic inhibitor. So at the end of the day, what you would expect is that compound X would show the same behavior as the 2DG low or the 2DG high dose. We grow the cells in 13C glucose, and we actually did a time course. We collected at 1, 2, 3, 5, and so many hours. And we only looked at UDP glucNAc. And we wrote a software, I will show you the link later, that actually was able to deconvolute the isotopic patterns of UDP glucNAc into the pathways. This is an example of the hexosamine, or the glycolysis-related molecule in UDP glucNAc. Showing the profile, what you see is actually the time and then the percent of incorporation. So per time point, you can see in the different conditions, so compound X, 2DG low, 2DG high, and the control setup, you see the incorporation into this subunit. And you already have like an idea that you can see that the 2DG and compound X are not showing exactly the same behavior. If we focus on the first hours at the dynamic phase, we actually see that indeed the compound is not really blocking glycolysis. Plotting the incorporation over time into this subunit and adding a trend line, you get these equations. And that's very nice, because now I can actually do an assessment of the kinetics. If the control is 9.6, so the AX is 9.6, we see that with the low 2DG dose, we actually have like 30% inhibition of glycolysis. With the high dose, it's almost fully blocked. But interestingly, the difference between compound X and the control are very close to each other, and actually we do not see at certain time points an actual difference. So at the end of the day, we could really conclude that this compound is not blocking glycolysis, it's blocking something else. And we did then find in follow-up experiments that it was blocking something in the pentose phosphate pathway. Another example is ATP. This is the molecule. If you look at its origin, there's the ribose there coming from the PPP. There's also the adenine coming from the serine lysine metabolism. And again, these large molecules you have to think of, look at the pieces of the puzzle. And imagine if you were a cell and you have to build this molecule, you have 2 options. Either you recycle these subunits, meaning that you take them from DNA or RNA degradation. That's a lot of bases and riboses that you can recycle. Or you're going to take subunits that you newly synthesized. But every combination is possible. It can mean that we're looking at a section of the ATP that contains a labeled ribose, but not a labeled adenine, but vice versa. We can also look at a part of the ATP that consists out of a labeled ribose and a labeled adenine. And it can also mean that we're looking at a non-labeled ribose and a labeled adenine. So all these combinations can occur. But that makes it very interesting to look at the system. And this is a collaboration with Brian Gonzalez from the UGent, where we're looking at ATP. So you see all the isotopes going from no incorporation to +1, +2 until +10. And what we did here is we grew cells, control cells in blue, starved cells in yellow, and high lipid content cells in pink here. You see also that we have the 12C control, and this is really to show you that the incorporation that we see is really pure. So we should never have higher than 2-3% incorporation in this 12C control, but we expect none. And then the actual setups. So if we look at ATP, and I think the most easy one is the +5, that is actually representing the pool of ATP only containing a labeled ribose. So in normal conditions, we see quite high percentages of this +5, around 35%. If we look at the starved conditions, there's very low amounts of this. And if you look at the high fat conditions, it increases again. Also take into account that the ATP pool was actually the same. The abundances were the same. So what we can conclude from this +5 present here and an absent or much less absent here, is that in starved conditions there's a much higher recycling. The cell will actually not invest in putting carbons into pathways and build molecules. It will be designed to be very efficient with the limited nutrients it has available. Also here for the +1, +2, which actually reflects the adenine synthesis, so the synthesis of bases. We see high percentages here. Again, in the starved conditions, it's much lower, reflecting that these cells are very efficient in recycling. So they're not going to do much de novo synthesis of nucleotides, and that also helps us explaining what's going on. If you would have just looked at the abundances, and that's very important, I told you the abundances were not changed. There's as much ATP in the starved cells as in the control and high fat cells. You would never been able to conclude this kind of information. So here at least we can say that there's a much higher recycling. So we're looking at where do these cells recycle the riboses and the nucleotides from, but also very important, can we actually provide them as a nutritional support? So and this relates to malnutrition, if we actually give nucleotides or parts of nucleotides, the bases and the riboses to cells, can we now also help recovering these, help in the malnutrition profile? This is the software that we wrote a while ago. It's called MAMES. What it does, it's basically taking isotope logs of composite molecules, and it converts it into numbers. For instance, in the UDP glucanac, you will have a number on glycolysis, a number on the Krebs cycle, a number on so on. And if you then actually compare two conditions, A versus B, you can actually then tell, like, look, my glycolysis dropped 10%, my glycolysis went up 200%. This is what the software does. Also, we had limited time here to actually cover all of the aspects of tracer metabolomics, but keep in mind we also have an applied course in 2025. This is an annual course that my team organizes. It's in Leuven, so if you don't know the city of Leuven, it's famous for beer, chocolate, and so on. And now also for our tracer metabolomics course, which covers many aspects of mass spectrometry, data analysis, data interpretation, and so on. And with this, I give the word to David. Thank you.
All right, thank you for your attention so far. So I'll take this last section and we can have lots of questions and discussion afterwards. I'll try not to step back and fall off the stage. So you've had a really great introduction to GC and LC tracer metabolomics from James and Bart, with a lot of insight as to why it's useful, particularly as a really good complementary technique to help interpret steady-state metabolomics and really peel away those layers to understand what's happening underlying those pool size changes. So this is, of course, James's slide that he described really beautifully, looking at the incorporation of carbon into metabolite citrate and the different aspects of how it could be labeled. So what I'm going to take you through is ways in which you can extract this data so you can make the most of it and interpret what is going on in all of these different types of situations. So we really are trying to get people to be encouraged to do this type of analysis. And there are a few challenges, but they're not insurmountable. It does require a bit of specialist knowledge when you're processing the data, and this is complicated by a few things. Presence of fully, partially, and unlabeled molecules in samples, which makes it a bit harder to ID your compounds, and Bart's already given you a few insights as to how you can overcome that and what you should be doing. You've got naturally occurring stable isotopes in nature, and they will be perceived there in your mass spectra, which you need to correct for to really get an accurate quantification of your label incorporation. And of course, once you've done all of this and gotten out your very useful information, you want to put that into some kind of context of visualization of labeling patterns and pathway mapping, as has been demonstrated throughout these last two talks, to be able to interpret what's happening in terms of your biology, and also ways to then present that and describe it to your audience. So not all software packages provide an end-to-end solution for addressing these challenges. And so for this workshop section, I'm going to go through considerations for tracer metabolomics experimental data processing setup, things to look out for and to be aware of, options for some data processing pipelines, particularly ones that we've had experience with ourselves, and just mentioning also a few new ones that have appeared in the literature, and then examples of ways to take these outputs and visualize them and present. So first and foremost, as has already been expressed, tracer metabolomics is a targeted approach. You really need to be having a good understanding of what it is you're analyzing so that you can manage the downstream steps to doing the data processing and getting your accurate label incorporation measurement. So, as we said, for accurate calculation of stable isotope incorporation, you must account for naturally occurring isotope abundance. To do this, the molecular formula of the metabolite ion that you're measuring or using as your representation for a metabolite is required to do this accurate natural abundance correction. So this is relatively straightforward for LCMS. As Bart has said, LC is quite nice and easy. You extract the samples after you've done your harvest and wash and extraction, and then load it onto the LC and run it. So your metabolites that you're detecting are there in their natural state. Whereas for GC, we're having to derivatize them, particularly for polymetabolite analysis, with some kind of derivatizing agent to enable that transfer into the gas phase. So you really have to take into consideration these groups that you're adding on and take them into account when you're doing your natural abundance correction. So just to give you an example of this, so I won't cover LC because it's straightforward, but for GC, this is what you would have to take into consideration. We have here citrate, which has been demonstrated a fair bit already, and I'll continue with this as my example for most of these slides. So you've got your citrate molecule there. This is not the best pointer. Which we will derivatize with trimethylsilane. So this is a silicon group with 3 methyl groups attached, and this derivatizes any of the replaceable hydroxyl groups. This molecule then has a total molecular weight of 480 atomic mass units. So with the GCMS, we're usually doing electron ionization, which results in fragmentation of this molecule before measurement on the mass spectrometer. The resultant mass spectrum is fantastic when you're doing profiling and you're trying to ID compounds. This is like a mass spectral fingerprint. But you then need to be well aware of what these different masses represent for when you're doing stable isotope labeling. So in terms of fragmentation, as I said, this full derivatized molecule is 480 atomic mass units. We see this mass of 465, which is essentially our molecular ion, the M-15, the loss of just one of these methyl groups to give you the 465 ion. This is the molecular formula for that ion when you count up all of the carbons, hydrogens, oxygens, and silicons. And we need to also be aware of how many carbons are capable of being labeled within that entire molecule. Another example, should you not be able to use the molecular ion, which is really the best thing that you can do throughout. Quite often the molecular ion is not very sensitive and sensitively detected, so you may need to choose another representative ion. So if we take something like 363, which is the M-117, so that's the loss of the carboxylic acid group and the derivatizing agent, you end up with 363 and this molecular formula and a possibility of 5 carbons that are going to be labelled within that fragment. And this is all very important for the downstream natural abundance correction and determination of fractional labelling. So just to demonstrate what this looks like over 24 hours, if you're labelling citrate, so citrate as set up the way, or an experiment set up the way Bart has described, you've got your cells in culture, the label media has been applied and they're taking up the label. Once you've stopped your experiment and extracted, you've derivatized and detected it on the GC. And as you can see, unlabeled, there's a whole lot of, so this is your unlabeled iron, and then a whole lot of naturally occurring background abundant mass isotopologues. But as you go into your time course, you see enrichment of +2, +4, etc., up to 24 hours, which is what we are now going to try and integrate all of these mass isotopologues and understand what that label incorporation is. And for the end, if anybody wants to come and tell me what mistake I've made on this slide, I'll be happy to hear if you've picked it up. All right, so coming into these challenges, it's already been mentioned, once you start labeling, it's very hard, or it can be very hard to ID some of the compounds of interest. So again, with citrate, the 465 iron, you've got a possibility of 6 carbons being labelled, and you can have enrichment all the way out to +6. So the unlabelled mass spectrum of your unlabelled, and then unlabelled or M0, and then M+1, M+2, M+3, so on, will become something more like this under labelling conditions. So this is an unlabelled mass spectrum up here, whereas it's labelled down here. So whether it's LC or GC, this kind of mass spectrum can become quite confusing if you don't know what you're looking at. This type of mass spectrum won't be matched in a matching algorithm in your typical software or library matching tool. So again, as Bart had said, it's ideally run an unlabeled sample. This helps you with compound ID, but also helps you know whether there's any artifacts that may look like labeling when in fact they're not. And you should also run standards to help with this compound ID. So then analysis then requires integration of all of the isotope log peaks. So again, 465 is what we're looking at, to then understand the representation of all the possible instances of label incorporation. So all of these things I've been going on about come to this point of correcting for the natural isotopic abundance. And here is a lovely equation for how to do this. Do not ask me what it means. And this is one of the challenges, and what perhaps scares people away is making sense of something like this to actually correct for that natural isotopic abundance. So as you can see, this is time 0. You've got your natural isotopic abundance here, and without correction, correction prevents overestimation of label incorporation. So this is why you need to do it. So 0 hours versus 1 hour of labeling, you can see that without correction, there would be overestimations of what that fractional labeling is. So something like this natural abundance correction algorithm should be then contained into a software that you're going to use down the track, and that's what I'll talk about in the coming slides. So having now corrected your data, there are a number of then outputs that you're going to get out of your labeling experiments. The absolute abundance, the isotope log distributions, as shown by both James and Bart, and of course the fractional labeling, what is the percent uptake of carbon into the metabolite pools. All of these pieces of information are super important, as has been demonstrated so far. And the question then is, how do you then express this information in ways that help you, firstly, interpret the biology, and then after that, help convey that information to others in a really nice, clear manner. So this data then can be expressed as tables, graphs, heat maps, and metabolite pathway type schema. So what are your data analysis options then? So really dependent on the type of platform you're using and the vendor from whom that platform is, there could possibly be vendor-based software options. So you could have peak picking and integration. So outside of a vendor, something like Elucidata, Maven, which Bart mentioned, and Poly, which is also from that company, which is a paid service, has this whole pipeline that you can use. That's great if it's something that you just want to start using with very little setup time and get some help to do it, but there are also a whole lot of other options. You can have the full pipeline from a vendor, and these are primarily LCMS solutions. The ones that I'm most aware of are pipelines within Thermo's Compound Discoverer and also within Agilent Mass Hunter VistaFlux. Again, they're good, but you're locked into that vendor software. The other thing is some of these softwares can be a bit black box, so you may not always know how exactly they're doing things like your background correction or some of the other methodologies. So that then brings us to open source software, which I'll focus on a bit more. So again, peak picking and integration, so Maven, as we've already talked about, or something like XCMS, and you could go into Poly again, but for a full open source pipeline option for LC and GC solutions, here are some of the options that you have, or ones that we've worked on ourselves. Bart's already described Maven to you really beautifully, and so I won't go into any more detail around that, but essentially it is a targeted methodology. You can put in your list of metabolites that you're looking for, adding in the information around the molecular formula and the expectation for the number of carbons to be labeled. This can then set up that you integrate the M0 and then all its mass isotope logs as needed. The output from Maven can then go into something like this isocorrector package that we've been using in our lab at Metabolomics Australia. The reason that we've gone with this package is that it allows for both single and dual isotope natural abundance correction. So we've talked just mainly about single isotope tracer metabolomics today because we've wanted to keep it a bit more simple, but this gives the option for this more in-depth analysis as well. It's been compared to other well-known programs, and including, I think, AccuCore, one of them is what Poly uses. So it has very similar capability. It's maintained and supported under the bioconductor repository, and it also has features for correction for both high and low-res instrument outputs. So for us, we've just integrated it into a pipeline within our lab. So the Maven output gets taken up. It's been wrapped in a shiny package. So with an interactive GUI that transforms the Maven data, runs the isocorrector package, and then displays the results. And so what this gives you is an output of a number of an Excel spreadsheet with a number of sheets within it, the natural abundance correction data set, fractional abundance and fractional labeling, and then we've also added in now a few little options for data visualization in the form of bar charts. This is just a mention of something I just saw more recently. I haven't had any experience with it, but just to say that clearly tracing metabolomics is very popular, and there's ongoing development work to build data processing pipelines to enable the research community. So this is simple. With this one, it uses an XCMS input for peak picking and integration prior to the natural abundance correction and then respective data presentation. So that's some LCMS options. So I'll move now into GC, and again, that's more of my strength rather than LC. DEXI is a software package that has been built within our lab at the University of Melbourne. It started off, so Michael Dagley is the person who's been writing the package, and we've been continuing to develop it. It was first published and released in 2018, so what's available out there is really quite old, and we've significantly updated it since then. It's currently not publicly available online, but if anybody does want to use it, if you're interested in GC data processing, come and talk to me afterwards. We're really happy to share it and get other people's opinions off it. The package will take in any GC data that's been converted to netCDF format, and it gives you the option for processing the data, visualizing the peaks from each file, its M0 and the mass isotope logs, and allows you to integrate that. And being in a targeted manner and knowing what you're looking for, this data processing actually becomes really quite quick as compared to completely untargeted profiling workflows. So it has the ability to show a number of different panes to give you views of the actual chromatographic data, as well as quick snapshots of the mass isotope log distributions, or a number of other options that we have within the pane view over there. So the main functions are manual integration to, again, make sure that you are picking and integrating accurately all of the mass isotope logs. Things like retention index support. We've also built in native support for both Agilent and Shimadzu files, because those are the instruments that we've used. We're also working on using MRM data as well, but that's something we can talk about another time. And then, of course, outputs into things such as Vanted for the visualization aspect of things. Another option for GC data processing is out of James's lab at the Crick. This is Manic. Again, another beautiful option. This gives you basically a survey mode of all the files that you're looking at, and you can get to look at them, integrate them all in the one go. Again, a very fast processing methodology, and it gives you insights into pool sizes and incorporation of label into the metabolites. So, as I've said, you've gone through all of this data processing pipeline to pull out fractional labeling information, isotope log distribution patterns, and absolute abundance. What are some of the ways that you can then make use of this data to start making sense of it and also conveying this information to others? So there's some simple ways to do it. You could just simply represent the fractional abundance just as simple bar charts. You can now actually also compare mass isotope log distributions between a control and a test and look to see if there's differences, or you can incorporate the two of these into the one type of representation. So then putting this into a pathway context, something like Vanted, which we have incorporated into DEXI but can be used as a standalone, you can choose, again, because it's a targeted methodology, to create your own little pathway diagrams and to map your data directly onto this. So for my lab as a facility, we're always working with biologists who are new to this area and also quite often new to metabolomics. So if you pass them a data set, which is just a sheet of labeled incorporation, it's really hard for them to put it into the perspective of what does that mean in a pathway sense. And so even just having this simplistic pathway map with the fractional labeling in that pathway context starts to tell the person a story about where there is a shift in the uptake of carbon from a given carbon source. So these are just very simple ways to start getting people to wrap their heads around what the data is telling them. So from Bart's lab as well is Travis Pye's. This is another way of representing the data. So now rather than just using a representation of the percent labeling of the pool, it also represents what the pool size is for that experiment. So in this case, with the experiment where the wild type and the test were labeled with glucose, there's a slight change or slight decrease in labeling under the test or the treatment conditions. It's also handy to know whether or not there was a significant difference in the pool size, which is represented by the size of the actual circle, so how much grey area has been shaded in there. So this kind of representation can also then be put into a pathway format. And I'm just taking a small little section out of Bart's paper here. But this is very nice. So this is an untreated cell line, or same cell line treated with antimycin A. And as you can see, that under labeling conditions with glucose, the treated cells, once you hit acetyl-CoA, there is little to no labeling, plus also a complete reduction in the actual pool of acetyl-CoA. This follows through into the TCA cycle. Again, untreated, you can see the flow of carbon going from 76% down to 48% and through the TCA cycle in the untreated, whereas citric acid is not only not labeled, but just not detected either. Whereas we see for some of the other TCA intermediates, while they're not labeled under antimycin A treatment, there is a measurable pool, which suggests that there is synthesis of these metabolites from some other source. And again, this type of information tells you to then go on to do perhaps another labeling experiment with something like glutamine to see what is happening to provide you with these other metabolite TCA intermediate pools. So you can also just represent things quite simply in terms of bar graphs representing the mass isotope log distributions, and this can be done as ratios. So a nice schematic here of the carbon flow. This is actually taken from a paper that I worked with a student working on prostate cancer, and they were looking at benign versus localized versus metastatic prostate cancer. So under glucose labeling conditions, they used a citrate to pyruvate ratio where +2 citrate labeling versus +3 pyruvate gave you an idea of pyruvate dehydrogenase activity, whereas citrate +3 versus pyruvate +3 gave you an idea of pyruvate carboxylase activity. And under benign versus malignant conditions, you could see a big difference in how these two types of samples utilized glucose and pushed it into the TCA cycle. We can see the same kind of example using with glutamine utilization in the same set of samples. And in this case, what you're looking at is uptake of glutamine into the TCA cycle. You have the entry of glutamine via glutamate into alpha-ketoglutarate, where it's 5 carbons, dependent on whether it is oxidative or reductive. So if it's reductive, you'll see the alpha-ketoglutarate going straight to citrate, and citrate would have a +5 labeling pattern. Whereas if you have the oxidative cycle, it'll go via succinate, come back around, and citrate would have a +4 labeling pattern. So again, you can express this just as bar charts and start to see that the metastatic cancer samples start to have quite a different utilization of glutamine compared to benign or localized samples. And finally, Escher Trace is a very nice piece of software that gives you very, very pretty but busy pictures. So this tries to put everything into one space, context of metabolic pathway, mass isotope log distributions, pool sizes, fractional abundance and labeling, which is great, but can be overwhelming for somebody that's new to this type of data and may not actually make it that easy to interpret. But again, it gives you so many options for how you can represent the data, and I think you can then pick and choose what you like to then convey your message. So just to wrap up this section, for data processing and visualization, understanding the underlying principles of tracer metabolomics will allow you to set up a streamlined pipeline. There are a lot of options for both vendor and open source data processing for both LC and GC experiments. And I'd say that data visualization, whether it's for metabolite profiling or tracer metabolomics, is an ongoing challenge. The methods you use are dependent on the end use case and what message you are trying to get across to people. So the more options that are out there, I think the better it is for people to know what they can use to convey their message. So you have the requirement for data interpretation, more complex, less organized, but all the options. Oral presentations, perhaps you might just want an overview with a broader picture to be able to describe to people. And for a publication, maybe you want something a bit more succinct and focused to support a specific point. So to conclude the workshop overall, we've really tried to convey that tracer metabolomics is something that's really achievable. Most people may think that it's in the same vein as metabolic flux analysis and flux balance analysis, and this can be quite intimidating. We really hope that you guys have taken what we've presented, and it's encouraged you to try out these types of experiments to get greater information out of your metabolomics experiments. So we've given you this primer on tracer metabolomics. We've tried to show it's less complicated than metabolic flux analysis and flux balance analysis. The experiments are achievable with informative data, and this can be even from optimization samples. I've experienced that before. It enables a deeper interpretation of steady-state metabolite profiling data and can provide another level of discrimination between cell states and types. I'd also like to encourage everybody, if you're interested, that there's actually an imaging and fluxomics session this year. As James said, last year there was one talk that mentioned stable isotope labeling once, and actually even with imaging there was no talk. So go to this session at least and see what people are doing. And I think one of Bart's team members is giving a presentation in that session. So with that, I think we have half an hour and we can take lots of questions. We can have a lot of discussions. People can come and tell us whether they've got better options than what we've presented up here. But I think it's great if we can encourage the metabolomics community to get into more tracer metabolomics. Are you, James and Bart, are you coming up here or? Because I look scared.
Yeah, so happy to take any questions if there are any. And if there aren't, I can always keep talking about stable isotope labeling and lipid patterns. Any questions on the floor just now? Yeah, we have one. Oh, yeah, sorry, the microphone's all the way down here.
One?
Yep.
Thank you. Really good workshop, really interesting. Most of the data here was from the cell cultures, right? And in the tumor field and the brain tumor, we see not that only different cell types outsource parts of the pathways, but also the different organs, like something happens in the brain tumor and then liver taking care of metabolism for the tumor. So what are your thoughts or your suggestions for the introduction of the labels in the whole body of the animals, like let's say mice for simplicity, not humans. Thank you.
Okay, yeah, that's a great question. So just in case anybody couldn't hear that, the question was, what are the things that you need to take into consideration when you're doing stable isotope labeling in whole organisms? And I'm assuming you're meaning a mouse model rather than a human model, but because that would probably be a bit too expensive to do. But yeah, so at the Crick Institute, we do a lot of cancer studies, and there's many different ways to go about this. But I guess there's two main considerations you need to make when you're doing carbon-13 labeling studies in the mouse. And that's whether you do a bolus injection, so that's doing one big injection of the metabolite at once. And there are ways of doing that, tail vein, intraperitoneal. Or do you do a transfusion, so it's a slow introduction over time? I don't have any ready slides to show you, but actually what we do see is that we can see quite big differences there depending on which option you take. And so it would be wrong of me to say one's better than the other. If you can do it, if it's right to do it, then you should maybe try both. But I think usually what people would do is some kind of initial bolus injection with a follow-up injection later on. So that's the first thing I would say that you need to take into account. The next thing you would need to take into account is how to harvest the organs, I guess. So that's really organ dependent. Quite often we'll be doing things that are multi-organ. So as with all metabolomic studies, the most important thing we need to do is quench the metabolism as soon as possible. One way that we like to do that is to have a designated order of organ harvesting after the cull, so that at least, even though that means that one organ is always going to have a more quickly quenched metabolism, at least it's consistent. Okay, so perhaps it might be the liver, then followed by the kidney or the lung, whatever. It depends on what you're particularly interested in. Again, there's a number of ways to do it. People have different ideas about that. The way that we like to do it is to, as swiftly as possible, drop it into liquid nitrogen. And at that point, what we would generally do is lyophilize it and grind the tissue. That can be in a bead beater or even a pestle and mortar, that's fine. I know other people still would then take the wet tissue, put it into a pre-chilled system, like a pestle and mortar, and then grind it while it's still wet. I don't like that, really. It might be worth talking about the heart as well. So the heart is full of blood. So when we do heart studies, labeling studies, the first thing we do is freeze clamp it. So again, we've got a freeze clamp. I can't remember the technical name for a freeze clamper, but it's literally a clamp that we put in the -80 or in some liquid nitrogen, get that heart and squeeze it. I will warn people that is not a pleasant and it can be a messy experience to do that in controlled conditions. I could talk about that. I mean, it gets more complex than that as well, of course, because one of the things we would want to do is to dissect away. Let's talk about liver tumors, for example. Tumor heterogeneity is such a hot topic right now. And so despite what I've just said about freezing as swiftly as possible, what might actually be a better thing to do would be to dissect away tumors from the adjacent tumor environment and then freeze and freeze dry at that point. But that's not really conducive to the multi-organ harvest that I just said. So again, I think it's taking your particular biological question and harvesting in the best way. Anything else?
Yeah, I've got a couple of things just from experience as well. So I guess the other thing that we haven't talked about at all is deuteration. So you can also do that kind of approach with animals. You could feed them to low level and then look for turnover. But coming back to the more polar metabolomics, the prostate cancer example that I had up there, those were actually PDX tumours on the mice. They were actually taken and sliced because they were going to multiple different analyses, and those freshly sliced tumours were actually put into labelled media baths and allowed to label for a period of time. So that was another option. And then coming to human subjects, we've actually been working with a researcher at the Peter Mac Cancer Institute in Melbourne who has gotten ethics approval to actually label patients. So she's a melanoma researcher, and again take this kind of bolus approach, and then when they resect the tumour, we're taking that and we'll be doing the analyses. So those are some of those options in the in vivo sense.
Okay. Just one thing, like when you do in vivo labeling from a biochemical point of view, brain is ideal. If you infuse 13C glucose, it's very fast labeled. However, if you then continue infusing, brain produces lactate, labeled lactate, goes into the system, and then you have 2 labeled nutrients in your system. So be careful with like harvesting multiple organs. It becomes quite complex as well, because after 2 hours you reach like quite high levels of other labeled nutrients that pop up, because organs are active and they are producing all kinds of labeled molecules then. So that is also like something that you have to take into account. But from a brain perspective, that's ideal because it goes very fast. At least 13C glucose goes fast into the brain, labels very nicely, so you can actually have very short time frames. So that limits the infiltration of other labeled nutrients.
Don't know. Have we got is there a mobile mic? Does that move? Sorry, I'll try and get. Oh, it does move.
Thank you very much. Nice talk. And no, I wanted just to say that on the section dedicated to fluxomics, I will present results for the in vivo studies on the heart tissue, so we can discuss in better then and see our experience.
Yeah.
Okay, thank you.
Yeah, certainly not straightforward. That's for sure. Okay.
I have a question. So thanks for the workshop and interesting things that we are sharing. I'm just, and thanks for all the challenges that you are showing about this type of experiments. And what about challenging? What do you think about the MS2 level of identification for the isotopologues? So in this software that you have presented, most of this software doesn't use MS2 level, just retention time, and MS level 1. So what do you think about that?
Yeah, I guess, yeah, we've not really looked at that because we are just going so targeted. And it's that thing of looking specifically at a known list of metabolites. So we haven't got into that at all, or on my side, but I don't know, Bart, if you've got some insights.
Well, MS2 level is very interesting, but you need to know where the fragment belongs in the molecule. And that is now, I think, the limiting factor. We see, like we try now in my lab with EAD, you see beautiful fragments, very rich fragmentation spectra, very nice labelings, but we cannot pinpoint where in the molecule these fragments are coming from. So I think that is now a limitation in tracers. So if that will be solved, that we have annotated MS2 databases saying this fragment belongs to that part of the molecule, it's for sure an interesting, I think also for fluxomics, very interesting to explore.
If you take a look on all the enzymatic reaction, you can know exactly what is the label carbon or nitrogen that is left in the fragment and not, or is loose. So looking at the biochemistry, but we need something that can do it automatically, not just following all the reaction of the specific pathway. Okay.
I was just going to say one little thing as well. So we have done a little bit of stuff like that, but with lipids mainly, and then things get very complicated. But but again, the importance of always doing a parallel study that's unlabeled, where you can do your identifications with that. You absolutely need that, especially in the lipid world as well. But again, happy to talk more at length a bit later.
Back to your cell experiments. You've shown a couple where you've done steady state and some kinetic analysis. Is that a majority of the experiments that you perform, or do you also do pulse chase or more exotic experiments? And do they have particular applications that they're suited for?
That wasn't exotic enough for you. So I guess some of the things that we haven't talked about, I don't know if it would be that exotic, but certainly mainly what we've talked about is using fully labeled carbon-13 glucose. And that's definitely not all that we do. And I don't think I've got a slide that will show it, but if you are quite clever and perhaps use glucose that's labeled in the 1 and 2 carbons, then you can get some really neat information on the activity of glycolysis versus the activity of the pentose phosphate pathway and how both of those contribute to downstream pools like lactate and pyruvate. So you can do some pretty natty things like that. We don't tend to do any pulse chase experiments, do you?
No, not. We are going to do one because, this is what the researcher was keen to try, and it doesn't typically make sense to do it, but it's interesting. So basically, they will be doing the thing of labeling and then switching back into unlabeled media and look to see the washout. So I've never done it before, so I was happy to go along with it to see what it is we're going to see and see what the value is to us. But it's not something we would typically do, and I guess especially in the context of my lab, people want to keep quite focused on what they're doing and minimise unnecessary experiments. So it's just a good opportunity this time. So in the future, maybe I'll have some info. Unless have you done that? No. Okay. Cool. No. I'll wait to find out soon, hopefully.
Certainly, carbon-13 glucose is the easiest thing to use. It's probably worth pointing out that carbon-13 glucose, when it's fully labeled, is not much more expensive than glucose, whereas carbon-13 glutamine is, maybe 500 times more expensive than glutamine. So even when we're doing experiments that are focused on glutamine metabolism, we will always start with a glucose labeling just because it's cheaper.
Yeah.
And
great way to optimize your setup.
And so to do all those sort of initial time course experiments that you'll always need to do, we always start in carbon-13 glucose. Again, we do work with nitrogen-labeled glutamine to track where that goes. And also, carbon-13 acetate can be very useful for looking at lipid labeling studies as well. And then, of course, I think one of them, Dave mentioned doing dual labeling as well. So if you can be quite clever if you want to be with doing that kind of work, especially if you've got a high-resolution mass spec where you can differentiate between a labeled nitrogen, perhaps, and a labeled carbon. So you might still have +1, but it'll be very subtly different that you'll be able to discern when you're using high-res mass spec. Certainly not a cheap MSD like I like to use. Any other questions? Oh, yeah.
When we are doing the natural abundance correction using isocorrector or any other packages, what we usually expect is like the labeled carbon percentages decrease, right? But what I have already seen is like the correction is also applied for the 12C carbon. Why is that so? Like, why should we actually apply the correction for even the 12C carbons, like M+0? Why not only from M+1? And second one is, so let's say I have a metabolite which is increasing in the knockout or the wild type, and I have a particular labeling, And can I now compare only the areas of only M+2 or one of the isotopolog and say that this is how, this is the pathway that the increase is happening?
Okay, so I think I understand the second one. I might need to, in fact, I better not move because I might need to get clarification on the first part. So your second question was, if you've got a multi-carbon molecule and you know that you're interested in the M+2 because it comes from a particular pathway, that you should only look at that for all your future experiments. Is that what you were saying? No, that's not what you were saying. Sorry, what? Yep. Yeah. Yeah. Okay, so I think, is that maybe more related to Bart's talk, where you've got different contributions from different pathways? Well, I think it would be dangerous to rule out other things, and also you're going to have all of that information anyway. I can't think of a way that you would easily only look at certain isotopologs. Yeah. So I think it would always be dangerous to do that direct comparison without taking all of the other isotopologs into account. But
yeah, if I may, so what I showed was there's much more behind it. So like if we say, so each isotopolog of UDP glucnac is explained by all the possible combinations of, so a +6 could be like a +4 in the adenine or in the uracil, and then the +2 of the acetyl. So all of these are in equations. And then we apply, I think my postdoc, it's like a heuristic approach to deconvolute everything. But at the end of the day, we do get confidentiality that the +6 is really coming from the glucose and not from a combination of. UDP glucnac has 16 equations that are all explained, so the solution to one is really quite unique from that perspective. So yes, we're quite sure that it's not a combo of other potential labelings. And then of course it's the biochemistry. The +6 label is, if you just look biochemically at the system, the chance that it's combined by this +4 and +2, it's true, could be. But it's mainly explained by the fact that glucose went via the hexosamine biosynthetic pathway. We can also do fragmentation, so we have done that on that molecule, and then you can indeed see the +6, I mean the actual glucosamine separately, and then we do, this is like what we can do in addition to these molecules. So, does that answer your question?
So the first part of the question, it was, why do you need to do natural isotope correction on M0? But does that question mean, why does the M0 change when you do natural isotope? So when you've got an unlabeled molecule, there's going to be some that is, so 1.1% thereabouts of all carbon is carbon-13, right? So that means that if you were just if you were looking at a molecule that has got no labeled carbons, there will still be some. That means that when you do the natural isotope correction, those will just be put on top of the pile of the unlabeled ones. So I know it looks like you're doing a calculation on the M0, but it's all of those fake labeled isotopes that are just contributing to that unlabeled pool. Does that make sense? I feel like I could have explained that better. Does that make sense? Yeah. Okay. Okay. Any more? Any more? Okay. Well, good. Well, thank you very much for all your questions and your attention. And I'm glad I didn't trip up then, which I was quite scared of. Okay, so yeah, we're all available all week. Dave's giving another talk later in the week, and Bart's student, postdoc? Postdoc is giving a talk later in the week. So do check them out. And
Bart's also going to tell everybody about why it's good to have a career in.
oh yes, and for those in the early career side of things, there's the early careers night tonight, where Bart will be center stage, telling everybody how they can be just like him, and he's really excited to do that. So please encourage him. Anyway, thank you very much for your attention. I hope it was useful, and yeah, we're here all week. Thank you.
Excellent.
I think questions.